My name is Dr. Erinn Muller, and I'm the Science Director of Mote Marine Laboratories' Elizabeth Moore International Center for Coral Reef Research and Restoration. I'm excited to share with you some really great news that we have at Mote. We have documented for the first time that one of our massive boulder corals that we have outplanted onto Florida's coral reef through the process of microfragmentation is now gravid. What that means is that this coral is producing eggs and sperm so that it will be releasing these gametes into the water column during the next spawning event coming up. Microfragmentation is really unique in the sense that it cuts through the biology of the coral. What we do is we take large boulder corals or brain corals that grow really slowly if left alone, and we fragment them into very small pieces that we call microfragments. We allow them to recover and grow in our land-based nursery, and then we outplant them in clusters onto the reef tract. These corals grow more quickly because of the microfragmentation process, and so we can actually grow large boulder corals to be about the size of a large dinner plate within 2 to 3 years. And because we now know that this size is reproductively viable, we know that this method is going to work for coral restoration because it now produces these corals that are reproductively viable that can seed the next generation out on the reef tract. Although this is the first time we've documented our gravid corals through the microfragmentation process, we have thousands of outplants on the reef tract today, and those corals are going to become reproductively viable within the next several years and are going to be the foundation for our coral restoration success.